Hello everyone, welcome to another new class of human physiology. In the last class, you remember we discussed about the first class of neurotransmission, where we tried to cover about like resting membrane potential, how the resting membrane potential gets attained in the cells, mainly in the neuronal cells. Then also we discussed about the graded potential. We also saw how the EPSP and IPSP works. And very importantly, we mentioned that whenever the neuronal cell attain this threshold potential, which was about like minus 55 millivolt, the signaling process that initiates inside of the like neuronal cells, which is also called like action potential. So in this class, we will see like how the action potential happens inside of the cells, and we will see also like how different type of ion channels are involved into it. So let us stay with it. So what concepts will be covered? So mostly, as we said, in this class we will cover about action potentials. We will see like what different type of ions are ion channels are involved in cases of action potential, and then further we will discuss about different type of refractory periods. Okay. So let us see one by one. So what is basically action potential or neurons like electrical signal? Action potentials generally are like a very rapid or transient electrical signal changes, okay, and that basically kind of happens across the neurons membrane. And whenever this rapid transient changes of the electrical potential happen, neuronal cells basically kind of transmit a signal, right, from like the action like hillock area of the neuronal cells. So basically, if we have a action, this is like the cell body, and we have the action hillock area. So basically, initially like we had this resting membrane potential which was about minus 55 millivolt, mostly in this action hillock area, and then whenever it attains the threshold like potential, so initially it was like minus 70 millivolt, which was the resting membrane potential. And then from the resting membrane potential, what we get like the IPSP signals, right, and from the stimulatory signals we attain about like minus 55 millivolt, which was about the threshold potential. So whenever the cell attains this threshold potential, a cascade of kind of ion channel movement happens, and for that we will observe like a wave of electrical signal across the neuronal cell. This is only called as action potential. And unlike the graded potential, action potential mostly all or none event. So either it will happen or there would not be any action potential. Basically, as we said, the initial threshold potential is minus 55 millivolt, and in the next few slides we will see that what would be the different type of voltages that would attain based on like the ion channels movement. So in cases of action potential, the first step or the first crucial step or the like first stages of the movements that happens, which is also called as depolarization or the influx of the sodium. So let us see here in this diagram how the depolarization wave basically creates. So what we said again, let us recapitulate. Initially the cell or the cell membranes has the resting membrane potential, which was about minus 70 millivolt, and then via like lot of IPSP signals or the stimulatory signals near to the action hillock area, the cell got this threshold potential, which was the minus 55 millivolt. And whenever this minus 55 millivolt threshold potential is attained near the cell, you see on the neuron or on the axonal area of the neuron. So basically these are the axonal area of the neuron, right. So on the axonal area of the neuron, there are lot of voltage gated ion channels. So what are these? So these are voltage gated ion transport channel, right, and you can see like what are this channel, basically sodium ion channel. So you remember in our ion transport class we have discussed different type of ion transport channel for facilitated diffusion, somewhere leaky port channel, somewhere like ligand gated channel, but these are the voltage gated ion channel. So whenever the cell attain this threshold potential, which is about like minus 55 millivolt, Let's consider in this ion channel there is like a gate or the door that generally stays closed. When it is closed, for example, initially cell had like this minus 70 millivolt of resting membrane potential, right? So at rest generally the cells will these ion gated channels will be closed, but whenever the cells attain inside this threshold potential or the minus 55 millivolt, these ion gated or voltage gated channels get activated. So if the voltage gated channels get activated, what will happen? The gate will get open up, right? The gate will get opened, and as you know that sodium concentration outside of the cells is higher and sodium concentration inside of the cells is lower. So basically what will happen? Diffusion will happen and sodium ions will move from outside of the cells where sodium ions were more to inside of the cell. So some of the sodium ions will basically come inside of the cell, and as you know sodium ion carry what? Positive charge, right? And as positive charge of sodium ion comes rapidly inside of the cell, the potential which was initially at minus 55 millivolt or threshold potential, you see the potential will rapidly change from minus 55 millivolt to plus 30 millivolt. Okay. So basically what happened, again let's recapitulate, that initially we had resting membrane potential which was minus 70 millivolt, and after that through the IPSP signal it got the threshold potential which was minus 55 millivolt. Now on the neuronal cell, mostly in the axonal area, we have lot of voltage gated channel. So whenever this voltage or the charge of the cells become minus 55 millivolt, these gates will open up, causing lot of positive sodium ions to come inside, and the charge will completely change from minus 55 millivolt rapidly to plus 30 millivolt. And you know what will happen? This charge will basically propagate, the charge will move from this area to this near to this second ion channel area. So initially how much was the potential here? Initially it was like minus 70 millivolt to like minus 55 millivolt, and now lot of sodium positive charge are moving slowly near to this channel. So basically from minus 70 millivolt, because of this positive charge, it will become like minus 55 millivolt, which is also called threshold potential. And what we just said, whenever this ion channel near it attain the threshold potential, these ion channels will open up, again sodium ions will come in, causing the positive charge build up. Again same way the potential will become from minus 55 millivolt to plus 30 millivolt. Same way this positive ion will move to the next channel. Again this channel initially which was at rest was minus 70 millivolt. Once some positive ions move, it will become minus 55 millivolt, and this is the threshold potential. At threshold potential, this third channel will open up, causing lot of sodium to come inside. Again this localized potential from minus 55 millivolt will go to plus 30 millivolt. Okay. So basically this way one depolarization wave will kind of generate. So this potential which eventually becomes from like minus 55 millivolt to plus 30 millivolt, and this movement of this potential will create a depolarization, depolarizing wave. Okay. And let's see what happens next. Okay. And one more crucial thing just to finish before we go to the depolarization, in this end point of the axonal terminal area, you also see there are like lot of calcium ion channels, right? And these calcium ion channels are generally closed, but whenever the cell inside of the cell attains this plus 30 millivolt, so whenever inside of the cells attain this plus 30 millivolt, the calcium ion gated channels get opened, causing lot of calcium ions to move inside. Now after few slides I will discuss what is the importance of this calcium ion, but just try to remember that this way the cells on the upper part of the neuronal cells, basically they are attaining like minus 55 millivolt to positive 30 millivolt, and a depolarization waves are created. And once this depolarization wave reaches to the axonal terminal area, it activates the calcium ion channel, causing calcium ions to move inside. The role of the calcium ion we will discuss very soon. Then simultaneously, so for example, what we just said in the last slide, let's consider we had this initial depolarization wave on top of here, causing lot of like sodium ion channels to open, causing sodium ions to flood in, and the voltage initially changed from minus 55 millivolt to plus 30 millivolt. Now let's see what happens on this down area of the neuron. Okay. So basically, so how much we initially got the cell potential? So initially it was 70 millivolt, right, at resting membrane potential. Now after some IPSP, after the IPSP it got like the minus 55 millivolt, which was the threshold potential. And after that what happened? Depolarization happened, depolarization. And what it caused? It caused the cell potential to become minus 55 millivolt to plus 30 millivolt, right? And at this 30 millivolt we said like this created like a depolarization wave. Now as the inside of the cells got this 30 millivolt potential, what it does basically, it inactivate the sodium ion channel. So in the last slide, as we said, like the minus 55 millivolt, when it is going to the next, it is activating the next like potential channels, but whenever it reach the plus 30 millivolt, automatically it deactivate the sodium or it inactivate that sodium channel. I will discuss little more very soon, but just remember, whenever the cell attains plus 30 millivolt, two things happens. First, it inactivate the sodium ion channel, right? Second, what it happens is activate various potassium voltage gated channels which are present in this area. Okay, so neuronal cells also has lot of potassium voltage gated channels. So at plus 30 millivolt of potential, this potassium ion gated channels or voltage gated channels get activated or open. So what will happen? Basically this potassium voltage gated channels will get opened at the plus 30 millivolt, causing lot of potassium ions to come out. Why? Because generally you know, right, potassium ions are more inside of the cells and potassium ions are low outside of the cell. So basically simple facilitated diffusion happens. So facilitated diffusion causes lot of potassium ions to move out from the cell. And what it would cause to the potential? So initially what we said, initially the potential was plus 30 millivolt due to the depolarization wave, and now lot and lot of potassium, a huge number of potassium ions which are positive in charge, they will basically move out from the neuronal cell and it will come outside. And as lot of positive charge are coming out from the cell, it will leave lot of negative ions on the inside of the cell, causing the potential to rapidly change from plus 30 millivolt to minus 90 millivolt. So you see such kind of rapid changes is happening here, and this wave which is generated like this ion potential changes from the plus 30 millivolt to minus 90 millivolt, this is also called as repolarization or repolarizing wave. Okay. So basically in the last slide we saw like how depolarization wave created, right? So depolarization which was about like minus 55 millivolt to plus 30 millivolt, and on the same time there is like on the very same time we will also see a repolarizing wave, we will also see like a repolarizing wave which would have like about plus 30 millivolt to like minus 90 millivolt. And basically both this depolarization and repolarization kind of happen simultaneously. There is not a significant time lag, and using this depolarization and repolarization wave, neurons basically kind of sends this transmission signals towards the terminal area and from like one presynaptic neuron to the postsynaptic neuron. Okay. So this is the overall kind of scheme of the thing, where you saw initially cells were at resting membrane potential or minus 70 millivolt. From there we got the threshold potential using the IPSP, and now on top of this neuron this depolarization wave got created and causing like the potential movement from the minus 55 millivolt to the plus 30 millivolt. and whenever this plus 30 millivolt attains here, it opens up the calcium ion channel, and as it opens up the calcium ion channels, lot of calcium ions come outside. Okay, and I will discuss what is the role of these calcium ion channels very soon in the almost similar time, because this positive 30 millivolt also activates or opens up the potassium voltage gated ion channels, and as potassium is high inside and low in outside, lot of positive potassium ion kind of moves out of the cell, causing a significant build up of negative potential or negative ion charge, which basically attains about like minus 90 millivolt. This is also a hyperpolarizing condition. This is also a hyperpolarizing condition because the cells eventually kind of move beyond of the threshold resting membrane potential. So you remember initially at rest, the resting moment potential was minus 70 millivolt, right, and then depolarizing wave caused like plus 30 millivolt, and now when the depolarization is happening, eventually due to lot of extra potassium ion movement, the cells like the potential movement cannot get stopped like at minus 70 millivolt, it eventually comes to like about minus 90 millivolt. So although this is called as repolarization, this excess negative potential that get attained by excess movement of potassium ions from the cell, this is can also called as hyperpolarization. Okay, hopefully like the steps of depolarization followed by repolarization is clear to you. Now briefly quickly discuss about this calcium ion involvement. Okay, so as you see, so one more thing very importantly, whenever the cell attains this minus 90 millivolts, so here is also like calcium ion channels you can see. So this minus 90 millivolt, what it will do, it will basically inhibit the calcium ion channel. So what will happen, no further calcium ions can move inside. So no further calcium ions can move inside. Now let us see what is the calcium ion role, right. So as you see, like there are a lot of like vesicles, there are a lot of this type of vesicles, they have what, they have neurotransmitter to it. So these vesicles which are present to the axonal terminal area, they have lot of neurotransmitter, and they can be like glutamate, right, they can be glutamate which are like stimulatory neurotransmitter, or they can be GABA which are inhibitory neurotransmitter. So basically cells, what it is trying, it is trying to kind of use this potential to open up the calcium ion, and this calcium ion has a specific role for the release of this neurotransmitter from the axonal terminal area to this like post synaptic junction, to this post synaptic junction, right. If I consider there is maybe one more kind of neuron here. So how the calcium ion is helping into it? So you see this vesicle has lot of proteins to it, and same way the axonal terminal area also has some protein. What this calcium ion does is basically this calcium ion creates like a bridge between this neurotransmitter vesicles and the membrane. So initially the neurotransmitter vesicles are kind of not kind of very near to the axonal terminal membrane area, right. They do not have any connection between the vesicles and the terminal area, but whenever the calcium ions come inside, this calcium ion kind of creates a bridge between whom, between the proteins that are present on the vesicles of this neurotransmitter with the proteins which are present in the membrane of this axonal terminal area. And whenever this bridging happens, what it does is like this bridging cause these vesicles to kind of pull near to this membrane, and whenever this vesicles get pulled near to the membrane, it basically gets fused. Okay, so basically this vesicle get fused and exocytosis happen. You can see that this process of exocytosis happen, and the neurotransmitter molecules basically removed in the postsynaptic junction area or between like the two neuronal area. So in this way calcium ion has a very important role because it basically creates a bridging between the protein of these neurotransmitter vesicles along with the membranes, and using this bridging they basically pull the neurotransmitter vesicles towards the membrane area of the axonal terminal site, and eventually via the exocytosis those neurotransmitter molecule which can be either stimulatory like glutamate or inhibitory like GABA, they are ejected or removed in the postsynaptic junction. And once these neurotransmitter molecules are removed, what will happen? It will again create a IPSP or EPSP signal in the next neuron. So in this way the signal will basically continue, and this whole process of this depolarization and repolarization wave followed by this removal of neurotransmitter to the next neuron is called as action potential, okay, which is called as action potential. Okay, hopefully it is clear to you. So basically this is the neuronal action potential in a diagram, and all these like ion channels are involved. So this is like a pictorial presentation. So now we will discuss two important things, which are the refractory period, and also we will try to represent the whole potential or the action potential sequences over a graph. Okay, so let's first go to the graph and then we'll discuss about the refractory period. Let's go to the graph directly. So before we go to the graph, one last time, so what was the depolarization wave? The depolarization wave was from the minus 70 millivolt to the plus 30 millivolt, right? You remember, due to like the opening up or the activation of the sodium ion channel, there is a generation of depolarization wave, which was like from initially from the resting membrane potential, which was the minus 70 millivolt. Eventually it goes to the threshold potential, which was the minus 55 millivolt, and from the threshold potential it goes to plus 30 millivolt, which is called a depolarization wave. From there, same way, when this plus 30 millivolt was attained, it opened up those potassium ion channel, causing the potential to drop from plus 30 millivolt to about like minus 70 to minus 90 millivolt. So up to minus 70 millivolt we can call it as repolarization, and as we know, like it doesn't stop at minus 70 millivolt, right? Basically because lot of potassium ions come outside, it goes via more kind of polarization, which is about like minus 90 millivolt, and this stage can be called as hyperpolarization. Now let's see the whole thing in the plot, where we have like the x-axis, which is the time, and the y-axis, which is the potential. So basically we have the y-axis, which is the potential, and x-axis is the time in millisecond. So where we are, let's see the different levels. This is the resting membrane potential, which was a minus 70 millivolt, right? And then this is the minus 55 millivolt, which is the threshold potential, right? So what happens basically, at rest cell is at minus 70 millivolt. Now we said what, like there are graded potential or IPSP, right? There are graded potential or IPSP. Basically it is the integration of different stimulation signal. If that happens, the cells from minus 70 millivolt, it touches the minus 55 millivolt, which is also called as threshold potential. Whenever the cell reaches this threshold potential, the action potential initiate. And how it initiate, you see in this like ion channel, where initially the ion channels so basically let's consider this is the sodium ion channel. So basically let's consider these were the sodium voltage gated ion channel, and each ion channel can have two gate, okay? Each ion channel can have two gate. One is activation gate, one is activation gate, okay? It can be called as AG. And then another is the inactivation gate, another is the inactivation gate or IAG. The second is the inactivation gate. So initially at rest you can see the activation gate is closed, right? Initially at resting membrane potential you see the activation gate is closed. But whenever the cell reaches this threshold potential here, you see what is happening, the activation gate of the sodium ion channel, which was initially closed, it got open, right? Whenever it reaches the threshold potential, which was a minus 55 millivolt, the activation gate which was initially closed, it basically opens up. See, the activation gate opens up, and whenever the activation gate opens up, slowly the inactivation gate also opens up. So what happens, along with activation gate, as it opens up, the inactivation gate also opens up. And when both the gates opens up, what it will do, it will cause lot of sodium ions to come inside of the cells, and not only the sodium ions will move from outside of the cells towards the membrane, and because these inactivation gates are also open, it will also facilitate those ion movement towards the cell inside of the cytoplasm. Okay, so due to this inactivation gate open up, the sodium ions will eventually comes inside of the cell, causing the cell membrane potential to rapidly rise to almost like plus 30 millivolt. So at this plus 30 millivolt, we said, and this is also called the depolarization wave. And what we said, what will happen at the plus 30 millivolt? At plus 30 millivolt, this will basically kind of deactivate or inactivate this inactivation gate, right? So at this point, this is the peak point. You can see this is the peak point. This is the peak point where the potential is about plus 30 millivolt. So activation gate is still open. The activation gate is still open, but this 30 millivolt basically inhibit this deactivation gate. So basically it will inactivate. So basically what it will do, it will inactivate this inactivation gate, causing closing of this inactivation gate. And what will happen here, because the inactivation gate is closed, as much as ion comes inside. So if you try to give more stimulation, more signal, more IPSP, as much as stimulation you generate, the sodium ions will eventually not able to come inside of the cell, right? Because this gate is closed, this inactivation gate is closed. So even though sodium ions can come inside of the ion channel, but the ions cannot come inside of the cell. The ions will not be able to come inside of the cell at this peak point. And now from this peak point, what we said, same way the potassium ion channels will get open, right? The potassium ion channels will get open because at this plus 30 millivolt, they get activated, the potassium voltage gated ion channels, and lot of potassium ions will come outside of the cell, causing the voltage to drop rapidly from plus 30 millivolt to about minus 90 millivolt. You see how the hyperpolarization is happening. So basically from the peak point of plus 30 millivolt, the potential will drop to the minus 90 millivolt. Okay. So eventually this initial step of minus 70 to plus 30, which is called as depolarization, then plus 30 to about like minus 70 can be called as repolarization, and from the resting membrane potential minus 70 millivolt to minus 90 millivolt can be called as hyperpolarization. Now let's see what are the two refractory period we have said, right? So there are two refractory period. One is absolute refractory period and second is the relative refractory period. So at this peak point, from this point, right, from this point to almost like 1/3 of this potential, as we said, as much stimulation you can generate, there cannot be generation of any further action potential, because this is already like the peak point, and at the peak point of plus 30 millivolt, the inactivation gates are closed. So even though some sodium ions come inside, they will not be able to go inside of the cell, causing no further neurotransmission. So this is the time lag or the refractory period, which is called as absolute refractory period, where as much as you create the IPSP or you give the stimulation signal, it will not be able to translate into an action potential. And whenever the cell or the potential reaches to the hyperpolarizing state at minus 90 millivolt, it basically takes some more time from the minus 90 millivolt to the threshold resting membrane potential, which is minus 70 millivolt, right? So this time lag where the cells eventually comes to the hyperpolarizing state, and from the hyperpolarizing state it goes up to the resting membrane potential followed by the threshold potential. So this time lag where you can give lot of stimulation signal, but it takes few extra fold of stimulation because it already came to the hyperpolarizing condition. But here if you gave extra stimulation of IPSP or stimulatory signal, eventually that summation or integration happens and the potential can eventually reach to the threshold potential. So this gap or this period is called as relative refractory period. But just remember, in cases of absolute refractory period, as much as you give more stimulation or IPSP, it will not be able to translate to an action potential because their inactivation gate is closed. But in case of relative refractory period, you can increase the few fold of extra stimulation that can translate to basically attaining to the threshold potential followed by the process of action potential. So hopefully everything is clear. We already discussed about the synaptic transmission. You can go through the notes here. Important thing is like there are few proteins you have to remember. For example, this type of V snares protein or synaptobravin, these proteins are present on the vesicles. So this type of snare protein like V snare or synaptobravin, these proteins are generally present in the vesicles. Same way there are proteins which are present on the membrane which are called as T snare protein or syntaxin. Okay, so what we just said, that in cases of the vesicles we will see some proteins which are called V snare protein or synaptobravin. You will see some proteins in cases of this membrane also which are called a T snare protein or syntaxin. And what we said, whenever this calcium ion channels open, whenever this membrane potential reach from minus 55 to plus 30 millivolt, the calcium ion channel moves and then this calcium ions comes here causing the bridging between these two protein like V snare and T snare, and whenever this bridging of the protein happens, they will pull the vesicles near to the membrane, and whenever it pulls near to the membrane, eventually it will fuse, and through the exocytosis process these neurotransmitter molecules will eventually come out. So this we already discussed. Go through the note. Hopefully you like the classes of like neurotransmission, both part 1 and part 2. If you have further questions, please discuss with us during live sessions. You can also drop your questions via email. So activity questions: how do drugs or diseases that affect ion channel function alter action potential propagation and synaptic transmission? You can also give some specific examples. So try to answer this question. Hopefully you are enjoying different classes of human physiology. Let's meet with you very soon with another new class. Thank you very much.